So let's pick up where we left off with the fungus, with the ascomycetes or the sac fungus. The name of these organisms, remember, I'm sorry, the groups that these organisms belong to, the ascomycetes and the groups we've looked at, remember, has to do with the type of structure they produce when they're engaging in sexual reproduction. That's how we divide them up. So the ascomycetes are named for the fact that during sexual reproduction, or the fruiting body that they create, looks like that little thing in the lower left, that red thing, it looks like a cup. So that's why these are called cup fungus, okay? This is the biggest group of 65,000 species, as we mentioned yesterday. The moles, the truffles, the morels. I think on the left-hand side is a morel. And remember I said that even though it looks a little bit, it doesn't really look like a cup. Imagine that each one of those little holes or divots in that organism is a cup, right? Ascomycetes or this Ascomycota, many lichens are of this group, typically in terms of the fungus. Remember, lichens are a mixture of organisms. We'll talk a little more about lichens today, but they're an interesting play on the word species, because they're actually two critters that live together that are very different from one another, but have lived together for so long that, I mean, this is the ultimate symbiotic relationship. So we'll get to that in just a moment. But about a quarter of the fungus species that are found in lichens, you see it's a green algae or a cyanobacteria, a prokaryote or a green algae, and then some fungus. And about a quarter of the time, that's going to be a sac fungus, a ascomycete. Reproduction in these organisms. All right, again, let's go ahead and start, since we're focusing on this idea of the fruiting body they create as a way for them being classified. So on the lower left, I've highlighted in red, you got an asci. So the asci are the parts of the organism in which the spores are produced, and they're contained in a fruiting body known as an ascocarp. All right, so there again, an ascus is a sac. So an ascocarp is the sac that is the fruiting body, right, of this type of a fungus. And each of the little finger-like things that you see there that are related to asci, those little things, if you want to back up on the circle just a little bit counterclockwise, those are little things in which inside is a picture you see there, a microscope picture. Those are where spores are being produced, all right? And we call those ascospores simply because they're spores that are produced by these groups of fungi, right? All right, so we'll go ahead and finish that circle here in just a moment, but let's go on with the asci. So the asci, spores are produced in the asci, and those are then dispersed, and if they find a place, they germinate and so on, with all the caveats that we've talked about before. And then if they do germinate, they start forming those long chain of cells. Remember that weird thing about fungus? It's about the nucleus. It's not really about the cells so much. But they form these long chains of hyphae, and these hyphae grow together in these huge mats known as mycelial. All right, so then if we have a mycelial a mat that engages in asexual reproduction, go off the left-hand side for just a moment, then we get just this constant production of asexual spores that are genetically identical. So what we're doing here is we're just populating the environment, right? Whenever organisms are cranking out things like asexual spores or when cells, like prokaryotes reproduce asexually, what they do is they're just cranking out the numbers. They're not so much worried about diversity in this case. Of course, that's how the prokaryotes get their diversity, is by cranking out so many numbers and playing the probability game. But you got these asexual spores, these things we call conidia, and that's a term we use to specifically refer to the asexual spores that these type of fungus, these ascomycetes, produced. Conidia, or these asexual spores, are produced on conidiophores. Phore, like sporangiophores, the thing that holds up where the conidia are produced. So, all right, conidia, asexual spores, conidia are produced, and they germinate and form hyphae, and that's the asexual route, right? Nothing really too unusual there. But let's go back to the sexual route for just a second. So then we get two different conidia, two different mating types. Remember, there aren't sexes in fungus, so we don't have a male and a female or anything like that. And it's going to be, mating types are going to be based on genetics. That's really what it is. It's two different individuals, two genetically different individuals. A plus and a minus of those mycelia will converge upon each other, and they kind of tie themselves up into a little knot with these organisms. And then that whole weird thing of plasmogamy and karyogamy with the time in between occurs. You remember I started off with yesterday talking about reproduction in fungus, and I said that, all cells do this when they're fusing, fertilization. It's all about the fusing of the cytoplasm, then the nuclei fuse. I mean, you can't do one without the other, obviously. It just makes kind of sense physics, right? But that what fungi do is they sort of play with that time in between when the cytoplasm fuses, the plasmogamy in this slide, and then when the nuclei fuse, or during karyogamy, when the kary meaning the nucleus. So that area in the purple, which is not an area that we've seen on other reproductive cycles before, wasn't in the alternation of generation. And of course, if you think about it, just in your own experience, it's not anywhere in the animal reproductive cycle. But this is where the fungi have found an alternative to being diploid, right? And I spoke about this yesterday, because during this stage in which they're heterokaryotic, or in this particular case, in this type of fungus, during this time between plasmogamy and karyogamy, they're dikaryotic, so 2 nuclei per cell, all right, in an ascus, and the ascus is the cell. You get some of the advantages of being diploid, right, because you have 2 sets of DNA, 2 sets of genetic information by which to be ready for the environment with, right? Don't forget all that. Tie that back to the evolution at the beginning. And in addition, during this time when these cells are dikaryotic or heterokaryotic, another thing that can happen is as these nuclei continue to come together in the chromosomes, they can continue to engage in genetic recombination. So it's crossing over. So they can cross over a bunch of times. And remember, crossing over is random each time. So the more times you do it, it's like shuffling a deck of cards, right? The more times you shuffle up the genetic information. So there's another advantage, if you think about it, having these cells that have more than one nucleus in it, that don't have a fused nucleus, that have 2 haploid nuclei instead of 1 diploid one. Nobody said it had to be a diploid one, right? It does seem that we find that there isn't some sort of an advantage to having dominant recessive alleles for the reasons we talked about in evolution. Plants do it that way with dominant recessive for the most part, so do animals. But when it comes to these organisms here, what they did is they said, all right, let's not go with 2 different alleles in the same nucleus. We're just going to have two different nuclei, each with a double set of genetic information. Right, so again, it's just this whole idea of playing with different ways to make variation work. Because remember, as I've said before, in the fungus or in the fungi, the focus isn't on the cell. It's not about the cell. It's not about the individuality of the cell. It's more about the nucleus. So for a very short period of time, the part that's in the pink here, for only a very short period of time do fungi spend their lives as diploid, right? After karyogamy occurs and they become that diploid nucleus, that zygote, they go right back into meiosis right away, generating variability, and producing haploid nuclei, right? 4 of them, as a matter of fact. Remember, meiosis, you always produce 4 cells. So 4 haploid nuclei, and then we're back to the beginning again. Those 4 haploid nuclei, each one of those divides, then you get 8, right? You get 8 ascospores. All right, so a little variant on the theme of this extension in time between plasmogamy and karyogamy. The same thing happened in the black bread molds that we spoke about yesterday. They formed that really gnarly-looking, rough-looking ball, that zygosporangium. And during that time, that was actually not just a dikaryotic stage. That was heterokaryotic. Let me go back and remind you and show you what I'm talking about. See, there it is on the right. So we get heterokaryotic. So again, we have this mixture of genetic information for ability to deal with environmental conditions. In addition, as I mentioned, the time for these chromosomes get together and engage in crossing over because of chemistry thing, folks. So if they're in the same area, they're going to keep doing that, and that's going to shuffle up genetic information. All you really want is variability, right? All you really want is mix it up. A lot of that variability may not work, but as we've talked, some of it might. Okay, the other major group of fungi are commonly known as the club fungi. Maybe because, many times folks think it's because they kind of look like a club. That one right in the middle, the red one, this is the one I think that was in the Alice in Wonderland stories where some cat was sitting on top of, if I remember right. But they kind of look like a club, but that's not exactly where they get their name. But a Basidiomycetes, a Basidio is a club-like thing. So, and I'll show you specifically why they're called Basidiomycetes, because you can notice in the picture here, while some of them look like the club in the middle, mushrooms, toadstools, there are these things called puffballs. I got one in lab I'll show you. It's just a loose sac, and you squeeze it, and all that smoke that's coming out is not smoke, those are spores. Like tons of spores, man. I'm telling you, spores being spread out again, tons and tons of spores, right? Variability in numbers, all that stuff. So they're not all look like clubs. There's some that grow on the side of trees, the shelf or bracket fungus. That doesn't look like a club. Really pretty ones, too, like that maidenvale fungus. Look, isn't that neat? That'd be a cool thing to see in the woods. And some of them aren't so pretty, like the ones that you see, the pathogens, oftentimes on plants. On the left-hand side, I believe those are maple with rust. It's not the metal, right? Your trees don't have iron in them by which to rust, but it looks like rust, but it's actually a fungus. And then on the right, that thing that's attacking the corn there is called a smut. So these things that are rusts or smuts are plant pathogens, so causing disease, and in some cases causing disease to the plant, but ending up being something that we can actually make use of, and I'll talk about that as we go through it too. So fungus, we eat fungus, right? And fungus are decomposers, so. All right, a lot of species of Basidiomycetes form mycorrhizae, those really important symbiotic fungus that live within the roots of the plants. These are very important wood decomposers. We've talked about how important that is. Lignin, that really a tough structure in trees that helps them, gives them their structure to stand up against the force of gravity. That's a really tough molecule, and there's only a few things out there, prokaryotes, and some fungi who can break down that lignin and recycle those nutrients back into the environment. So they're very important here. Okay, let's take a look at the reproduction in these folks. Again, let's start with the thing that gives them their name. On the right-hand side, you see something that I've labeled as a basidiocarp. So we had an ascocarp. Okay, now we have a basidiocarp. And again, it's a misnomer, but if it helps you kind of remember the major members of this group, think about the toadstool as being a club, and basidio means club. This is, again, the fruiting structure. That's where the carp comes from. The ascocarp was the sac-like fungus, and it was the ascocarp was the sac. This is the club. But again, it's actually more at a microscopic level. So hold on just a second before I talk about that. These toadstools, these basidiocarps, for reasons that I talked about early on, can grow very rapidly. Remember, the way the hyphae are connected, oftentimes it's just one cell or just a few cells, and maybe just an open cell with some of those septate in there. But the whole idea is you can move cytoplasm very quickly through that stuff. So what happens is, remember, most of the fungus, especially in this species, is underground. This is the whole thing with that giant organism, that 3.5-mile fungus I was telling you about. So all of that liquid can be flooded to the ends of those hyphae very quickly when conditions are right, causing them basically to balloon, right, to bulge up and balloon up into these basidiocarps, come up from underground subterraneanly underground, no kidding, and form a toadstool. Well, as I was saying yesterday, remember, like, sometimes you walk out in the yard after it's rained and you go, wow, those mushrooms came up quickly. They were there already, right? They're just hyphae said, oh my goodness, great condition, bloop, and they just flooded liquid cytoplasm down into the end of those hyphae and caused them to plump up like a balloon and form these things sticking above ground. And the reason they were sticking above ground is because we're going to get spores passed this way. That's the whole point behind doing this. if you flip open your typical toadstool, and remember, not all basidiomycetes are toadstools. If you flip open that basidiocarp, you see the gills on the bottom. And the gills have lots of surface area, and I don't need to go into that. That lots of surface area is going to allow you to create lots of spores, billions of spores. And the name basidiocarp, again, doesn't come from the fact that they look like toadstools, because they all don't. It comes from the fact that if we look down on the gills underneath the microscope, and we'll be able to do this, we can see these little lumps, these things called basidia. Those are the clubs. That's where the names come from. Look up in the upper left. I've got a microscope picture. This is very close here in the upper left to what you guys are going to see when we look under the microscope in lab next week. The blue lines are actually the edges of the gills, and the lumps that are sticking out are the basidia. So those are where the name comes from, the basidia. Now notice this the basidiocarp, the toadstool in this particular member of this particular group, the basidiomycetes, and the basidia and all that. Notice where they are in this in the life cycle. They're in that heterokaryotic stage. Notice basidiomycarps, we call them N plus N, not N. The cells are not N or 2N. They're N plus N because the nuclei have not fused. They've stayed together, right? All right, so now inside those basidia is where the N plus N is really happening at the detail. So you got a blue and a red mating type again, okay? Plasmogamy, when the mycelial of the two mating types come together, you see, so I'm going to back up a little bit on the circle up here. So we back up to how those basidiocarps are created. So mycelia of different mating types, a plus and a minus, come together. When they fuse, they form dikaryotic mycelia, the one that each has a blue and a red dot inside. And then those begin to grow much more quickly than the haploid mycelia of the parent. So you notice the mycelia that's got both the nuclei in it is growing out much more quickly than the one with only one nuclei. So eventually those dikaryotic mycelia outgrow all of the haploid mycelia, and those are the things that are popping up above ground. Those dikaryotic mycelia have cytoplasm flooding into them, and this basidiocarp, or this toadstool, pops up above ground, okay, in this heterokaryotic stage. Then karyogamy occurs, and again, we get a very short-lived zygote stage, a very short-lived diploid, very different than in animals, right? Diploid, we spend most of our lives diploid. Most of what we are is a diploid thing. We have parts of us that are haploid, but most of our bodies, our multicellularity here, is diploid. These guys are diploid for a very short period of time. Fuse the two nuclei together. This is all has to do with variation, folks, right? Meiosis is variation, fusing together is variation, and then those diploid nuclei undergo meiosis, produce 4 haploid cells again, 4 haploid nuclei. I guess I should say nuclei, since I'm trying to remember that we're in fungus. Inside the basidium, so there's 4 haploid nuclei. Each of those are different because they've undergone meiosis, and now in the sac fungi, those haploid nuclei divided by mitosis, so we got 8. But basidiomycetes, it doesn't work that way. You get 4 basidiospores, right? Basidiospores, ascospores and ascomycetes, basidiospores and basidiomycetes. But again, they're the same thing. There's haploid cells, and they go out and they germinate, and then hopefully find a place to germinate, becoming haploid mycelia, and the cycle repeats. This is sexual. Asexual reproduction does occur among these organisms, but it's not well known or not real common. So we don't put it in the cycle here. But again, it would work very simply, where they would produce asexual spores that would germinate back into other asexual spore reproducing structures. Here's a little picture in the bottom left. It's a electron microscope picture. So the basidium it looks like a club. In the basidium, the basidiospores are hanging out the club there, if you'll notice. The sterigmata, this word right here, that's the little connecting part right there, the little thin part, the part that connects the club, the basidium, to the spores. We always say that down microscopically at this level, one of the big difference between basidiomycetes and ascomycetes is basidiomycetes, these guys produce the spores on the outside. All right, so keep this picture, this electron microscope picture in your head for just a second. So they produce the spores on the outside, whereas in ascomycetes, right there, notice the spores are produced on the inside. See, the ascomycetes or the ascospores, in the ascomycetes, the ascospores are produced inside the asci. The asci are the finger things, and the spores are produced inside, whereas in the basidiomycetes, the finger things are now basidia, and the spores are produced on the outside. Why am I making such a big deal about this? If you're looking under a microscope and you were a mycologist, somebody who studied fungus, and you said, okay, well, let me see the spores. I see spores are being produced on the inside of these little finger-like structures, then it's an ascomycete. If it was spores produced on the outside, like I showed you in the other picture, like this one here, then basidiomycete. Right? See how that works? Okay, now back to this idea of how these things pop up so quickly. Like I said, sometimes after a rainstorm, you might walk out in your yard and see all these toadstools that popped up from everywhere. And not only you may notice that, but you might notice that they're also oftentimes sort of arranged in a circle or part of a circle. I don't often see it completely a circle. Now, historically, back in time, we called those things fairy rings because nobody seemed to know where they came from overnight. They popped up, so it must be something like these mythical creatures would come down to Earth at night and dance around in a circle, flittering fungal dust or something like that, and that's where the mushrooms would come from. Well, obviously, that's not the case, right? It's a nice story, kind of interesting. But now you have a much better way to explain it. What happens here is, let's go back in time for a second. These are in a circle for a reason. A spore, a basidiospore germinates, say right here. And the mycelia begin to grow out from it. Mycelia grow everywhere, branching, right? And they're growing out, and what they're doing is they're roaming through the environment, digesting nutrients. I should do the thing from Jaws here. And as they're spreading out, if the nutrients are equal throughout the soil, which let's assume they are, and it looks like the picture from it is, so we're underneath the kit here. See if I can draw that as a dashed line. The mycelia are going to spread out at an even rate, aren't they? So at any given time, if you were to draw a circle connecting all the ends of the mycelia, you would find that more or less it's in a circle. Now, rain comes, conditions come, and this fungus decides it's time to reproduce. It's time to produce spores. It's time to produce basidiocarp. So it takes cytoplasm and it starts flooding it to the ends of the hyphae, or into the hyphae. When it gets to the ends of the hyphae, all the areas I've connected with the circle there, it plumps up. So the reason why you get fairy rings in a circle, well, the reason why you get these toadstools popping up is because the mycelium mat underground, and the reason why it's in a circle is because from the point of origin, a spore, the mycelia grow out, assuming, again, the nutrients are equally spread in the soil, in a circle at an even rate. Now, I bet you some clever mycologist has figured out a way to determine growth rates, mycelial growth rates, by calculating the amount of time it took from a spore to a particular radius for a fairy ring. I wonder if he could look at this and say, yeah, that spore's probably germinated for, say 3 weeks now or something like that. That would be neat. Knowing by the size of the circle, right? The larger the circle, the longer the spore's been germinating. Speaking of fungi in our environment, they do all kinds of things, as I mentioned. They're decomposers, take care of cellulose, which is a big problem for us animals. Lignin, which is a big problem for pretty much everybody except the fungus and the prokaryotes. And the whole idea behind decomposers, folks, is recycling. Taking those essential nutrients, essential elements, carbon, or carbon-based life forms. A lot of carbon in lignin. If it was tied up in lignin forever because nothing broke it down, that would be a lot of carbon lost. And there's other things, nitrogen, potassiums, trace elements, those sorts of things. And decomposers are really, really good for, obviously really essential for environments, for recycling that back into the environment. Remember, that's what I was talking about yesterday with the problem in tropical environments, that the decomposition rate is so fast that there's a lot of decomposers, because those are great places, for decomposers to live in warm, moist conditions, that the stuff is turned around, recycled, and generated so fast that a massive amount of plant growth that goes down there sucks it up so quickly. And that's why the soils don't have they're not very nutrient rich. We say it's like a residence time. The amount of time the nutrients stay in the soil until somebody needs them is very short, very high demand and low supply, really. So if you think about it in business terms. Fungus are also good mutualists with plants, mutualists with algae. That's the lichen thing that I was talking about. We'll get to lichens again in just a moment. Blue-green algae, cyanobacteria, they are mutualists with animals. They engage in all of the symbiotic relationships we've gone through. This is similar to the chart that I showed you before. Obviously, the repetitiveness is because you have different species and pluses and minuses, and depending on how they're arranged. Mutualism is on there. I guess the only one I would add actually is predation. I would consider predation a plus and a minus, like parasitism. Predation is technically a symbiotic relationship. Some of these, of course, as we've talked before, are obligate symbiotic relationships. Some are facultative. We talked about that when we were discussing the prokaryotes and being aerobic or anaerobic. So obligate symbiotic relationships are those that organisms have to survive, and facultative are those that they can take it or leave it. Good examples of obligate mutualistic fungi are things like mycorrhizae that we talked about. Those plants need that to survive, right? 90% of all vascular plants have mycorrhizae. There are fungus that live inside a plant cell. Endophytes. Endophytes is a term we use to refer to something that lives outside plant cells, fungus, or it could be a bacteria. If it's a fungus, it's usually one of those cup fungi again. But you can see that picture. Those are the hyphae of the endophytic fungi living inside that plant cell. These are found in all plant cells. They can provide, in some cases, they actually provide protection of plant cells to herbivores, right? Plants can't fight back if a herbivore, a plant predator, comes through the environment there. What they can do, they can't scratch, they can't run, can't bite. They can make it. It can make you not want to eat them, right? For example, you get sick, or there's some toxic material that causes your tongue to swell up, or something like that, so that you want to stay away from that plant. So that would be important, and pathogen protection as well through maybe the toxins they produce. The toxins produced by the fungus actually protects the plant from things coming munching on it. And this would be a symbiotic relationship, right? Fungus can develop a symbiotic relationship really cool with animals. There are those that help in the digestion of guts, like the bacteria that we learned about with the cow. But these leafcutter ants, oh my God, they engage in this thing called fungus farming. What they do is they cut off these chunks of these leaves with their mandibles. They cut them all off, and they carry them over this fungus, and then they feed the leaves to the fungus. So they put the leaves obviously in, the fungus. It's not like it's got a mouth, but they put the leaves in the vicinity of the fungus. The fungus digests the leaves, and then after it's done, it produces these very nutrient-rich tips at the tips of its hyphae, and then the ants come along and nibble off the tips. Doesn't destroy the fungus completely, but these nutrient-rich. So basically what the fungus does is kind of condense these leaf nutrients together, and then the ants eat it back again. Isn't that kind of neat? So, we can have these really bizarre, so I was talking yesterday about meta-evolution kind of things that go on in behavior between organisms. Some of these, like I said, are facultative. Some are obligate. Lichens are an example of an obligate relationship between two organisms. There's about 15,000 species of lichens, but notice I put the word species in quotes here, or not quotes, in italics. The reason why is what a lichen is really challenges our idea of our definition of species, especially when we think about what Ernst Mayr called it, right? Reproductively isolated, blah, blah, all that stuff we talked about, the way we talked about how we identify species genetically, and all those things we've discussed so far. Now we're going to kind of look at a little curveball. A lichen is an obligate mutualistic relationship. In other words, it's necessary. It's mutualistic for both of the organisms involved, both partners, because if you tear one away from the other, they will die. They have to have each other survive. They depend upon each other for survive. They've evolved that way throughout the years. They may not, chances are they weren't that way to begin with, but throughout the years, a fungus, again, usually a cup fungus, and a photosynthetic organism, could be a prokaryote like a cyanobacteria or a green algae, or could be both in some cases, something that photosynthesizes, live together. Like look in the picture on the right, the one in the upper right. The fungal hyphae are the purple, the algal cells are the light blue. And the algal cells live in amongst the fungal hyphae. And what the fungus does is it protects the thing that photosynthesizes from light. Now, yes, it photosynthesizes and it needs light, but it's a matter of attenuating, just having the right amount of light. So the color that you see in a lichen, the reds and the greens and the blue-greens and the colors of some of the lichens, that comes from the fungus. And the color is a filter to filter out, just to get a right amount of light so that photosynthetic partner can photosynthesize without having too much. Drying out, desiccation, the fungus can also protect the algal partner from drying out. And of course, the benefits from the photosynthetic partner are obvious, right? The fact that they photosynthesize and can produce food. Now, in spite of the fact that this seems like a tenuous kind of relationship between these two very different critters, remember, if it's a cyanobacteria, we're talking about a prokaryote and a fungus, a eukaryote, that have lived together. And as I said, we call these things species. They have species names. But in spite of the fact that it looks like a tenuous relationship, oftentimes lichens are what we call pioneer species, species that are I think we talked about this when we talked about plants moving out on the land. Pioneer species are the species that they're often some of the first that move in. And what that means oftentimes is they are good at dealing with harsher environments than other organisms. And very often, as I mentioned, when I did originally talk about this, pioneer species actually change the environment chemically or et cetera, and making it better or easier, more conducive for later species to move in. It's that meta-evolution thing. So lichens, in spite of what they look like on the surface, actually can be found in some fairly harsh climates. We find a lot of them here in Florida. They're really good at living in hot conditions like we would deal with here in Florida, things that would challenge an organisms to deal with those kinds of environments. High sunlight. Been found in the fossil record since 420 million years ago, so they've been around for quite a while. Indicator species. An indicator species is a term that ecologists generally are going to be referring to. When we look at an environment and we want to know if that environment's being disturbed, and that could be for a variety of things, chemically, heat. In other words, some parameter in the environment is being upset. It could be human beings, could be natural, whatever the case may be. Oftentimes, there are one or maybe a handful of species that tend to be the first to show the stress. It could be a plant, could be an insect. For example, we could say we understand that when in these environments, when the oxygen levels get low, the first species that dies off is this larvae of some insect. So if you were wondering whether oxygen levels were dropping in a lake, and if you wonder why that's important, well, fish, that's important for fish, obviously. But if you were concerned about oxygen levels are dropping in a lake, you might say, all right, when you see these critters start to die off, when you sample and you notice, wow, there's a lot of dead of these particular insect larvae, or there aren't very many of them, then that's an indicator that something's wrong. Lichens are very often that way. So an ecosystem or an ecologist would, if he noticed the lichens dying off in an ecosystem, he might say, hey, there might be something going on that the trees and everybody else and the animals aren't showing yet, like there might be pollution or some chemical getting into the environment that's causing this. We'll talk a little more about this later on too, because this is the root of what we call the what's been the canary in the coal mine. I don't know if you ever heard that expression, but miners used to bring canaries into coal mines years ago to let them know if the poisonous gases were building up in the coal mines. So that's another indicator species, right? Sensitive to environmental disturbances, usually the first to show. There's a drawing down below again, algal cells and the fungal hyphae. The Ceridia, those little chunks that break off, those are for asexual reproduction, and they are just little chunks, a little bit of fungus, and some photosynthetic partner goes off on its own. Again, the whole species thing is weird, right? Because these guys are very different from one another, yet they can't live apart. Now, in your field notebook, I want you to identify 3 species, one lichen of 3 different types. What I mean by type has to do basically with overall shape. There are 3 basic types of lichens. There are those we call fruticose, foliose, and crustose. And it's very easy to remember them. The crustose ones, there's another picture in the lower left that might help, are crusting. They're encrusting lichen. On the trees here in Florida, sometimes it looks like a spotch of paint. Those are crustose lichens. I'll show you some in lab. The foliose ones, folio is leaf. Like a portfolio is a collection of papers. Think of papers as leaves. So a foliose lichen has a leaf-like shape or texture to it. There's one in the lower left and one in the upper right too. There's also another foliose one in the upper right, maybe a little better one right there. Okay, and then the other one, fruticose, is referring in the picture up above to this thing. Fruticose are shrub or bush-like. You look on trees, next time you're near a tree, you see lichens growing all over. If you look real closely, you'll see some real crusty, little tiny mini, almost like bushes growing on there. Those are fruticose lichens. And here's some common Florida lichens. Up in the top and on the left, deer moss, not a moss. Remember, be careful with that. Deer moss is not a moss. It is actually a lichen. So that stuff all over the ground, that blue-green color, deer moss or reindeer moss, is actually a lichen. The red is the Christmas tree or the Baton Rouge lichen. That's a crustose lichen. You see that a lot on the trees, like on campus. And then that Xanthoparmelia, the foliose one down below. In that particular case, if you got that for your field notebook, I don't need a species on that. It's hard to tell apart, but that's a foliose lichen. So that's going to be sort of paper-like, if you think about it. All right, so that's lichens. Let's talk a little bit about fungi as parasites, fungi as pathogens, and then we'll finish up talking about some uses for fungus. About a third of fungal species are parasites or pathogens. And if you think about their lifestyle, that makes sense, right? They're decomposers. They're used to living on organic material that is decomposing, that perhaps is already dead. So when Bambi falls over dead in the woods, it's the decomposers like the fungi that move in first. So it makes sense that they would, in many cases, be parasitic. They're living on something else, or perhaps even pathogen, disease-causing. Big problem with fungus, of course, they are for animals, but they're a bigger concern for crops, especially food crops, where we lose between 1/10 and 1/2 of the world's fruit harvest every year to fungus. And if you've ever grown any little garden, you know that that is a problem, losing your crops to fungus and disease. For example, you see there in the lower left the fungus that's attacked that ear of corn. The name of that fungus, there's a scientific name, but it's got I guess a common name, Withlacoochee, which reminds me of Withlacoochee, the river up by Gainesville. But Withlacoochee is actually a fungus that's growing on the corn, but it is actually a delicacy for human beings, right? I understand, I had never had any, but it actually has a very good taste, and it's used in many dishes in areas of the world in which this thing affects the corn. So. Interesting. We eat fungus, right? Mushrooms, those are all fungus. Tar spots, we talked about rust and tar spots on fungus. Ergot on rye. Ergot is the fungus. The name is Claviceps purpurea. This is all right here. This stuff grows on rye, which has been eaten by human beings. And this particular ergot that grows on rye, this Claviceps, as it begins to decompose the rye wheat, it forms a hallucinogenic material. So back in the days of burning the witches at the stake, what people have looked at, historical forensic folks, if you look at it, if you think about it that way, is many times what was seen as these symptoms of dementia, of being possessed and all that sort of stuff, had to do with folks that were eating rye wheat back then that was tainted with a hallucinogenic, like an LSD kind of material. It was causing them to actually have these hallucinogenic fits or trips, and folks saw this some signs of dementia. Burned some of them at the stake. Horrible, right? So some of these fungus can be, toxic to human beings, although some are delicacies. Some grow on our food that we eat. So that's something. The chestnut blight, chestnut trees. We don't have, I think there's just a few chestnut trees left in the United States. They used to be thick. I guess the old saying was that a squirrel could practically walk from, New York to Virginia, from one chestnut tree to another. They were so thick. The picture down below, chestnut blight, which is a fungal parasite, was introduced in New York City in about 1910, somewhere around that time. And what you're looking at in the map is the spread of the disease, causing about half to almost all of the chestnut trees to die by 1940. It spread out to halfway through the state of Tennessee. And now most of the chestnut, like I said, I think there's only a few. Remember the other day I said trees and groups are called stands of trees. I think there's only a few stands of chestnut trees left, actually. Fungal infections, one of the problems with those affected, they can spread very quickly, as you can see in this particular case. Frogs, amphibians in general, about a third of the world's population has been touched by this particular fungus, by Tracheotrichum dendrobatidis, called chytridiomycosis. It is a chytrid. That was one of the groups we looked at. You can see in the picture of the dead frog. It causes this huge decline in amphibian populations, leading to the decline or the extinction of somewhere about 200 species. There's the map. It's spread throughout the world. Spreads rapidly again, just like the chestnut blight. If you look down in Mexico, first reported in the early 70s, up around Mexico City, quickly spreading down into the Isthmus of Panama within just a couple of decades. If that doesn't strike you as problematic, the amphibian population declining, let me remind you that everything's all connected together. And that in our own backyard, one of the things that amphibians do for us is consume a lot of mosquitoes. So in general, a huge imbalance of an ecosystem like this by the wiping out or the decline of one group of organisms can no doubt have probably some unintended consequences on ecosystems. Sometimes these are natural things that move through the environment. Sometimes these are things that are made worse by human beings, in terms of moving things around and spreading disease, that sort of stuff. We also suffer from fungal infections, mycosis, animal diseases. Some are opportunistic, which means that they don't really cause a problem unless for some other reason your body's immune system is on edge or has been compromised. Candida, that's Candida albicans down below in the microscope, is a yeast, and it causes thrush or vaginal infections. And Candida and pneumocystis jirovecii, there we go, jirovecii, causes pneumonia in AIDS patients. Now, pneumocystis is a relatively rare form of pneumonia. Candida is really only usually a problem in females with vaginal infections, if you've ever had that situation, after taking antibiotics. You see, the antibiotics that you take actually deplete the good bacteria in the regions of the body where these Candida yeast would normally grow. So the Candida yeast are there, but in a normal body without the antibiotics affecting the makeup of the prokaryotes, the Candida is kept in check. But the antibiotics kill off the beneficial prokaryotes, and the yeast infection then begins to grow. That's why I'm being opportunistic. Pneumocystis jirovecii, the pneumonia and AIDS. Pneumonia is a symptom. It's the filling of lungs with fluid, which causes you basically to die from suffocation. Pneumocystis is a rare form of pneumonia because it's not a particularly pathogenic form of pneumonia. Your healthy immune system is usually able to fight it off, but someone with AIDS doesn't have that, right? So a lot of times infections, whether fungal or otherwise, are opportunistic. They're not a problem unless your immune system is otherwise compromised. A lot of fungus are that way too. Athlete's foot is a fungus. Ringworm, there in the lower right, is what ringworm looks like. That little inset of a picture is the microscope of ringworm. Ringworm is not a worm, but a fungus. It's actually an ascomycete again. And nail fungus. A lot of time when folks get older, they get problems with nail fungus. You see all the ads on TV. You know what I'm talking about. But those things are problematic for humans. But not all fungus are bad. Obviously, some of them are good. They're used in the production of cheese, alcoholic beverages. We already talked about Saccharomyces cerevisiae, which we use to ferment carbohydrates. There it is in the picture. Bread is obviously, we use that in terms of, that's a good one, bread. Yeah, I guess in the making of bread, we can use fungus, right? Cheeses, blue cheese, that sort of stuff. Antibiotics, penicillium. There's penicillium. That's a neat picture based on, we did our Petri dishes. There's Staphylococcus that's been spread on that dish, and then penicillium, which is a fungus, is growing right there. And notice the big thing about the picture, the part where they're calling the zone of inhibited growth. That's the light bulb that the fella who understood that penicillin had antibiotic properties, that's what he saw. I don't know if you remember the story. He was growing something on a Petri dish and left for the weekend. Penicillin mold, which is found in the environment. That's penicillin in the upper right, by the way. That's what it looks like. It's very cool. It's got like the Medusa head woman there. But he comes back after the weekend and he notices on his plates that the bacteria are growing, but wherever the penicillin is, there's this exclusion zone around it. Come to find out that penicillin has antibiotic properties, right? So it's that idea in science that chance favors a prepared mind. If you had the forethought to look at that and go, wow, that's something interesting, I wonder what's going on, then you might be the one we were discovering or talking about who discovering what penicillin antibiotics do. Yeast can also be used in genetic research. Why? Because it's simple genetically, so we can use it as a simple model. We've used it to do genetic engineering. Genetic engineering basically where you create recombinant organisms. Recombinant, again, is just a word that means that you change the DNA. So we could, for example, take a yeast cell, and I talked about this back with prokaryotes too, with E. coli and molecular cloning. You could inject into a yeast cell the genetic information on how to produce insulin, right? The stuff that's obviously important to be regulated in terms of diabetics, but not something a yeast cell uses. But as the yeast cell would read its genetic information and use its ribosomes to create materials, it would create insulin and not use it. We would just, same thing it does with alcohol during anaerobic fermentation, right? It produces the alcohol as a byproduct. Human beings take advantage of that and use it as a beverage, right? Okay, so that's it for fungus. I'm not going to go into the animals. We'll start the animals next week. If you have any questions, give me a